for the invitation. As you can see, I'm from NUI Galway and I'm the School of Chemistry at NUI Galway. I suppose we have a few people, I just selected out just a few people in the School of Chemistry. I suppose myself, my own background, I'm a synthetic chemist really. I mean, I make molecules. We make natural products. We actually do chemistry with natural products, I suppose, if you could define it that way. We work a lot with carbohydrates and chemical transformations of carbohydrates and using carbohydrates as starting materials in synthesis. I should say that about a year ago, we appointed Denise Tazdemir in the School of Chemistry. And Denise is a professor in marine biodiscovery, and her expertise in natural products chemistry as defined in terms of isolation of natural products from marine resources. This is her speciality. And structure determination. She's actually the Beaufort PI, so this was funding provided by the Marine Institute. She is the head of the Marine Biotechnology program in Ireland. So basically, isolation and structure determination of natural products from marine resources. And she's also working on the Nutrimara project, which is related to food applications, and she's working with seaweeds and so on, which many of you talked about today. So we have Denise in our school, if anybody wants to talk to Denise about possible applications. In terms of I will also mention Andrea Axleben. And Andrea is not a natural product chemist, but she is a crystallographer. And for example, if you can get single crystals of natural products, you can determine structure. That's one approach to it. We have, for example, Rob Woods, who's half-time between the University of Georgia and NUI Galway. And Rob works a lot with carbohydrates. And developing of influenza inhibitors, for example, is a project. He's a lot involved in modeling of antibodies and all these kind of things. In terms of facilities in chemistry, as I mentioned, we have X-ray crystallography. We have just installed a 600 megahertz NMR with a cryoprobe. So when you are, for example, determining if you have very small quantities of natural products, actually having a cryoprobe is very important because you get a lot of extra sensitivity, and that can help you with your structure determination if you don't have a lot of sample. We have other instruments as well in NMR, and also quite state-of-the-art mass spectrometry and LC mass spec techniques available, and expertise as well, from people such as Denise. And we have also technical staff in the school who provide support. So those facilities can be accessed, for example. My own area, as I mentioned, I am a synthetic chemist predominantly. But we have, most of our program has been linked in some way to bioactive compounds. I suppose natural products are, generally considered to be a source or the best source of bioactive pharmaceutical type compounds. I think in anti-cancer research, over 60% of the anti-cancer medicines are either natural products or they are closely derived from natural products themselves. These are just some. For example, we have worked in the last few years on natural products like this, like migrostatin and analogues of the natural products. They're anti-metastatic, potentially anti-metastatic compounds. They are macrolide structures. This is a related acyclic structure. These are other kind of macrolides we've been involved in. Racemic acid lactones. These kind of compounds have anti-angiogenic, anti-cancer agents. We have been working a lot with amino sugars. These are close analogues, structural analogues of normal sugars. They are, for example, glycosidase inhibitors. I think they're actually found under the skins of potato, in potato skins, for example. Compounds like these have been clinically approved for diabetes because they inhibit glycosidases and glucosidases and the breakdown of starch. We have been involved in glycolipid chemistry recently, synthesis of bacterial glycolipids, which are very potent immunostimulants. And not involved in peptide synthesis per se. This is a structure here, somatostatin. This is a natural hormone. What we do here in this area are mimics of the natural products, actually using natural products as scaffolds. We try to develop what are called peptidomimetics. So this is just the first slide, just overview. So we use, actually what we have done is we have used the amino sugars, which are natural products, to generate peptidomimetics. So in the case of somatostatin, for example, side chains on the peptide are a very important part of the pharmacophore. And so for example, the indole and, well, the tryptophan and lysine side chains contain the pharmacophoric groups. And using synthetic chemistry, we can graft them onto other natural products and look at their bioactivity. So for example, we have been doing that kind of chemistry using the amino sugars, which are, you know, I suppose it's a kind of a creative kind of activity. In terms of the synthesis, to make the amino sugars, we do chemical synthesis. For example, we can start from D-glucose and we can eventually make deoxynigeromycin. We have expanded that, for example, to produce things like castanospermine, which have been of interest in the clinic as anti-HIV agents and so on, and anti-tumor agents over the years. We can produce these compounds like deoxynigeromycin from things that are very available, like L-sorbose, which I think can come from biomass, for example. We can compare, we can take amino sugars, we can compare them with normal sugars, like, for example, manosamine, and we can create new scaffolds, again, based on benzomacrolactone type of natural products, and we can compare one type of peptidomimetic versus the other. So for example, you can see here that this kind of potency of these compounds is better than the other types, for example. And in terms of, if you want to get patents, I mean, we filed a patent on these kind of compounds, and we got, a very good report in terms of novelty and so on. So if you want to get patents and things that might be of potential interest to pharmaceutical industries and so on, you can do that. Other areas, say microstatin, these natural products themselves. This is a macrolide, a 14-membered ring-containing macrolide, which has this glutaramide side chain, which was produced in streptomyces, that was found to inhibit tumor cell migration. Actually, it turned out that much simpler analogues, when the synthesis was done, people were screening analogues and intermediates and that kind of thing that were produced by the synthetic routes. And much simpler analogues turned out to be about 1,000 times more potent in vitro. And so we don't have this glutaramide side chain. In actual fact, they're a lot easier to synthesize, for example, if you want to produce them biochemically. Whereas this one was the most potent, the macrolactone. It turned out that this macroketone and macrolactam here, and actually macroether eventually, they are active in vivo. They have been tested in mice models for metastasis, and they have shown activity, for example. So you can generate analogues of natural products which could be potentially more interesting than the natural products themselves. We have been involved in generating novel compounds. We have had, based on microstatin and dorigasin A, we have produced them from carbohydrate substrates. We have identified bioactive compounds. We've done the work, for example, in collaboration with the gastrointestinal unit at the Mater Hospital in Dublin. I used to work in Dublin and UCD for 11 years before I went to Galway. We have looked at, in terms of with the Centre for Colorectal Disease at St. Vincent's, these kind of compounds, they have used tumor explants from patients with advanced stage 4 colorectal cancer, and you can investigate the effects of the compounds, for example, on, say, what effect do they have on angiogenic factors like VEGF, that kind of thing, and they do show activity in those assays, in those kind of assays. We at NUIG Galway, Corrado Santa Canale, we are looking at the evaluation of the compounds in tumor cell models. Like, aside from the microstatin type compounds itself as well, we have prepared a range of mimics from readily available natural products. I suppose from glucuronic acid we prepare these kind of simple, relatively simple natural products, or macrocyclic compounds. And actually we kind of got interested in that because there was a paper from the NIH which showed these compounds from quinic acid were also inhibiting tumor cell migration. So we had a lot of experience with glucuronic acid, and we've actually prepared these compounds, and they actually do show activity. And it is believed that they might be related to these natural products called isomicrostatin, for example. So again, these would be kind of new structures, new types of inhibitors. So I suppose we get inspiration from natural products to generate maybe non-natural structures. I suppose that's kind of like what chemistry we did here. Relatively simply, you can prepare, we kind of came across making these macrocyclic 8-membered containing, sugars with carbohydrates embedded into them, and the compounds for screening, for example. And you can look and you can find natural products themselves which have been isolated, which are not a million miles away in terms of structure. Relatively simple to prepare, could be potentially interesting in terms of biological evaluations. I suppose we have been involved in cost actions. We were involved in a cost action on angiochem, which is related to angiogenesis. This group involved in angiochem have a new cost action now called STEMchem. So basically they're interested in molecules which could influence stem cells, especially cancer stem cells, especially drug resistance to cancer stem cells is apparently very interesting. So we had made macros, we had these kind of natural products here like apothymycin and so on, are inhibitors of a range of kinases which are very important in angiogenesis. And so we have been doing kind of structure activity, synthesis of analogues of these compounds and doing kind of structure activity studies. And we had collaborations through the cost action with Jim Lawrence at the University of Bergen. And so we have European collaborations, and there are other people in the cost actions who have interest in natural products. And as well, these kind of bacterial glycosphingolipids, they are found in bacteria, Sphingomonadaceae, in particular bacteria that don't contain lipid A. They're structurally, it turns out, quite closely related to KRN7000 or alpha-galactosylceramide, if you've ever heard of it. It's a very potent immunostimulant and has been in the clinic. And there is a lot, but it hasn't been successful in the clinic, and there is a lot of interest in developing glycolipids which might, and there are a couple of compounds going into the clinic now with structural variations in the lipid chain here. We had a lot of interest in glucuronic acid and galacturonic acid, and especially alpha. I think actually alpha-galacturonic acid are found in pectin cell wall structures, and we actually are quite interested to try to make those kind of things. But we have made the glycolipids, we made the lipid precursors from galactose. We had a new enomerization methodology in terms of generating the alpha linkage that you need here. And we've produced the glycolipids, and we have since produced a variety of other ones even more efficiently than what I've shown you there. So I suppose our expertise, well, we have at Galway, isolation and structure determination, which is linked to screening and often driven by screening, biological evaluation. I suppose our expertise in my own group is synthesis of natural products, and actually maybe chemistry or modifying natural products using chemical methods. I suppose I kind of sat down and thought, well, where is there potential for natural products? Well, number one, I suppose, if to generate the natural products themselves, particularly if they're not very available from natural sources, I suppose that is a challenge. But it's one possibility is to use chemical methods or other methods to generate the natural products themselves, or natural product-like materials even, which are needed for further studies or further applications. There's, I suppose, opportunities for chemical transformations of, let's say, readily available natural products to provide new substances for study or application. And maybe that includes producing higher value products from biomass, because you have all these, I think, I read a US report, there's about 30, chemicals that are readily available from biomass with a lot of chirality inbuilt, which could be very useful. Applications, they're chiral materials, I suppose. There's a wide application potential, whether it be materials or whether it be chemicals or pharmaceutical synthesis or chirons or whatever, using natural products. I suppose they are sustainable sources of chemicals for industry. And you kind of wonder, is there new possible applications where they could be used, particularly when there's a need for renewable resources. I suppose there's always the traditional screening of natural products and chemical biology type applications for natural products for screening and for biological studies and so on. I suppose we use natural products as well as an inspiration. So, I think there was a study done in the Journal of Organic Chemistry published a few years ago, which said that, most of the compounds actually that have been produced over the years, millions, are based on a very small number of scaffolds, like 5 or 6 membered rings or 7 membered rings. And there's a lot of opportunity, that people haven't looked enough at different frameworks. And that maybe is one of the reasons why drug discovery has kind of run into problems. So maybe there are needs for new frameworks. I suppose they come from natural products or maybe inspired from natural products. I suppose natural products are considered to be biologically validated, that they've been around for millions and billions of years or whatever, and that a high percentage of them are closely related to natural products. I'd just like to thank the people who have funded our research through the years, the HEA's Programme for Research in Third Level Institutions, Science Foundation Ireland, who have funded programme grants for me, the Irish Research Council, as they are known now, who were formerly IRCSET for funding PhD students down through the years, and Marie Curie Actions in the European Union for funding postdoctoral researchers, I suppose cost and industrial collaborators. We've had funding from Eli Lilly, Roche, and Astellas down through the years. And just say thanks for your attention.